This deep dive explores the dawn of base editing as a transformative force in modern medicine. Base editing rewrites a single letter in the genome, aiming for precise correction without cutting the DNA strands. The CPS1 deficiency case centers on a rare metabolic disorder that traps ammonia in the body and damages the brain. KJ Muldoon Jr., born with CPS1 deficiency, faced a life-threatening condition in infancy without a workable therapy. A daring 6-month sprint united researchers from Harvard, Massachusetts General, and multiple biotech groups. AI-enabled design allowed them to bypass lengthy animal testing and rapidly lock in a base editing strategy for liver disease. The patient's bespoke drug, kayjayguran, was manufactured across North America in a cross-continental relay. In February 2025, the FDA granted expedited approval, a landmark moment for personalized genetic medicine. By Christmas 2005, KJA walked for the first time, marking the first truly rapid path from diagnosis to treatment in under a year. Aurora Therapeutics emerged to scale such genetic miracles, led by Kiran Musunuru and seasoned executives. The company's mission targets NBO1 therapies as scalable platforms, starting with phenylketonuria and CPS1-like conditions. Base editing evolved from CRISPR-Cas9, a molecular scissors that cut DNA and sometimes cause unintended changes. The new era began with 2016 to 2017 work by David Liu, who introduced base editing to improve accuracy. Early milestones included A/B development and preclinical correction of mutations in mice. Unlike double-strand cuts, base editors chemically modify bases, reducing unintended genomic damage. In CPS1, the problem is an abnormal single base that stalls the liver's ability to process ammonia. Delivery to the liver uses lipid nanoparticles to ferry the editor to the target cells. The KJ case proved that a carefully designed base editor could be both safe and effective in a living patient. This milestone sparked a broader shift toward platformized, scalable genome editing therapies. The story is set against a backdrop of global biotech milestones and competitive market moves. Verve Therapeutics' high cholesterol program, VERV-102, demonstrated the commercial potential of in vivo editing. Beam Therapeutics pursued both in vitro and in vivo strategies, advancing sickle cell and liver metabolic diseases. In parallel, public institutions and universities formed dedicated gene editing centers for pediatric cures. ARPA-H announced programs to fund precise gene medicine and manufacturing improvements. China's biotech scene joined the race with domestically developed editors and rapidly advancing clinical translation. Companies like Zhongyi, Possible CS101, and YT101 pushed gene editing in blood disorders and cholesterol. Regulatory innovation followed suit, with NEJM editorials endorsing the plausible mechanism pathway for rare diseases. The plausible mechanism pathway enables approvals based on mechanism readouts and real-world data instead of large trials. Yet delivering editors remains the bottleneck, with delivery vectors mainly targeting the liver. Safety concerns persist, including bystander editing and long-term risks that require decades of follow-up. Cost remains a challenge, with initial therapies approaching landmark prices before efficiencies scale. Nevertheless, the KJ case proves that personalized, rapid therapeutic development is possible. The field aims to generalize from N of one to N of many through standardized platforms and regulatory pathways. In summary, base editing marks a turning point in medicine, shifting from bespoke miracles to widely accessible therapies. Looking ahead, continued AI-guided design, delivery innovations, and real-world data will accelerate progress. Key timeline milestones include 2016, Liu's base editing, 2017, ABE, 2019, preclinical, 2020's expansion to vectors. The video now pauses to reflect on the human impact beyond the science. We must balance ambition with safety, access, and ethical stewardship as the field matures. The CPS1 case remains a highlight, showing not just a medical fix, but a potential new model for rare diseases. As platforms emerge, more patients may benefit from engineered therapies designed around common disease mechanisms. On screen, the journey is far from over, with ongoing trials and new editors reshaping the therapeutic landscape. In conclusion, we witness a historic shift toward precise, programmable medicine that could redefine healthcare. This has been a deep read of MIT Technology Review's 2026 top breakthroughs focused on deep tech and trends. Thanks for watching this 3-minute overview. Stay curious about the next frontier in life sciences.